Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. In our previous lectures, we completed the multiple choice question related to the nucleic acid chemistry and basic molecular theory. Today, we'll move to the last part of biochemical reagents, assay development and design. Now let's focus on the question related to the assay development and design and review molecular biology ASCP questions from the previous exams. Let's go. Okay, the first question we have today. What is the primary goal of developing a diagnostic assay in molecular biology? Diagnostic assays are designed to detect specific nucleic acids or proteins, enabling the identification and quantification of pathogens, genetic mutation, or biomarker in sample. All right? Okay, the next question. Which of the following is the crucial step in the design of nucleic acid-based assay? The correct answer is designing specific primers or probes. Actually, designing specific primers or probe is essential in nucleic acid-based assay as they ensure the accurate detection and amplification of the target sequence. All right, let's move on. Okay, the question here. In enzyme-linked immunoabsorbance assay ELISA, what is the purpose of the secondary antibody? Actually, the secondary antibody in ELISA is designed to bind the primary antibody that is already attached to the antigen. Okay, it's typically conjugated to the enzyme which catalyze the colorimetric reactions, enabling the detection and quantification of antigen-antibody complex. So the correct answer here is to detect the primary antibody. All right, let's move on. Okay, next question. What is the rule of standard curve in quantitative assays? All right, the correct answer is to determine the concentration of unknown samples. Actually, a standard curve is created by plotting known concentration of target molecule against the corresponding assay signals. It's used to determine the concentration of unknown samples, as I said, by comparing their assay signals to the standard curve. All right, let's move on. Okay, next question. Which method is commonly used for high throughput screening of potential drug candidates? All right, the correct answer is microarray analysis. These techniques is high throughput methods used to screen thousands of potential drug candidates simultaneously. All right, so it's allow for the parallel analysis of gene expression or other molecule interaction on a large scale. All right, let's move on. Okay, the next question here. What is the primary advantage of using a multiplex assay? The correct answer is simultaneously detection of multiple targets. Actually, multiplex assay offer the advantage of detecting and quantifying multiple targets in a single reaction, making them efficient and cost-effective for comprehensive analysis. All right? All right. In the context of assay development, what does the term limit of detection refer to? Okay, let's define the limit of detection first. The limit of detection is the lowest concentration of an analyte that assay can detect with the confidence. That's the critical parameter for determining the sensitivity of the assay. All right. So the answer here is the lowest concentration of analyte that can be reliably detected. All right. Let's move on. All right. Which of the following is the common challenge in assay development? All right. A common challenge in assay development is ensuring the reproducibility and accuracy of the result. Okay. This involves optimizing assay conditions, minimizing variability, and validating the assay to produce consistent and reliable data. All right. All right. The next question. A positive control in assay is used to. A positive control in assay is used to ensure the assay can detect a known target. All right, let's move on. Okay, next question. Which step is the critical during assay optimization? Assay optimization is multifaceted process, but one of the most critical steps is the temperature optimization. Okay, this step ensures efficient primer binding and enzymatic activity, which is the crucial for improving assay performance. All right. So the correct answer here is testing multiple temperatures. Okay, next question. Which factor is the most critical for reducing false negative in a diagnostic assay? The correct answer is high sensitivity. High sensitivity ensures the true positives are detected and minimizing false negatives. All right. Please don't forget to subscribe to our channel to receive all the latest lecture. Okay, let's continue. The next question. What is the purpose of a spike and recovery experiment? Spike and recovery. All right. The purpose of a spike and recovery experiment is to check if the assay is accurate or not. Okay, how we'll do this? You add known quantities of substrate you are measuring to the samples. All right. This method help you determine whether the assay can accurately measures and recover the expected amount of the analyte or not. If the assay recover the known amount correctly, it demonstrates that the assay is accurate. Okay, let's move on. Okay, the question here, during assay optimization, which parameter is least likely to affect analytical sensitivity? Which one of them? The correct answer is substrate color. Okay, as sensitivity depends on the factors like incubation time, substrate concentration, and pH. All of these influence reaction efficiency. Okay, where the substrate color is just a visual endpoint and does not affect the sensitivity. Okay, the next question here. Which blocking agent is least effective in reducing non-specific binding in immunoassay with the high background noise? Which one of them? Bovine serum albumin, non-fat dry milk, gelatin, or casein? Let's see the correct answer. The correct answer is gelatin. Gelatin is less effective than bovine serum albumin, milk, or casein in blocking hydrophobic binding sites, especially in complex samples. All right? Milk and casein contain diverse protein that better minimize non-specific interaction. All right, the next question here. Cross-reactivity in assay primarily compromises which performance characteristic? Actually, this cross-reactivity occurs when the assay detects structurally similar molecules. This leading to false positive and reduced specificity. So the correct answer here is specificity. Okay, let's move on. Okay, the next question here. Which assay design features minimizes the carryover contamination? The correct answer is by using uracil N glycosylase. This uracil N glycosylase or UNG is enzyme that keep DNA and RNA in check by removing uracil residues. As we see in the image here, this DUTB containing fragments in DNA produced during the previous PCR reactions are broken down by UNG. So this helps prevent false positive by ensuring only newly sensitized DNA is detected. Alright, the next question here. A low signal in a chemoluminescence assay could result from the correct answer here is inadequate washing. In simple terms, if you do not wash thoroughly in the lab experiment, leftover substrates can interfere with your results. This interference known as the background noise. Okay? So this interference can make it difficult to distinguish the actual signals you are looking for. So proper washing ensure that only the desired signals are detected, leading to the clear and more accurate results. Alright, the next question here. Which parameter is evaluated during assay precision testing? The correct answer is interrun variability. This interrun variability measures how much results differ between repeated runs of the same experiment. Okay, so this helps us to assess the reliability and precision of assay by comparing results across multiple repetitions. Okay, the next question here. In CRISPR-based assay, what component provides target specificity? Okay, the correct answer is single guide RNA. Okay, in simple term, the single guide RNA act like a GPS that guide the Cas9 enzymes to exact location of the DNA that need to be cut. So, ensuring accuracy and specificity in the process. So the correct answer here is single guide RNA. Next question. Matrix effects in the assay can be minimized by. All right, the correct answer is diluting the sample or using matrix matched standards. But let me explain it in the simple terms. Actually, matrix effects are like unwanted noise in your samples that can mess up your results. For example, interference from salt or protein in the samples. So, by diluting the samples or using similar standards can help reduce this noise, leading to more accurate measurements. All right, let's move on. Okay, let's see the next question here. In ELISA, the purpose of watch step is to. All right, the correct answer is remove unbound reagents and reduce backgrounds. Actually, washing eliminates unbound antibody, antigen, or enzymes, preventing non-specific signals and improving accuracy. All right, let's move on. All right, the next question here. Fluorescent quenching infrared or fluorescent resonance energy transfers occurs when. The correct answer here is that the donor's emission is absorbed by the acceptors. Actually, fluorescent resonance energy transfers involve the transfer of energy from the donor dye to acceptor dye when they are close together, which reduce the emission from the donor. All right, as you see in the image here. All right, the next question here. Proton effect is characterized by. Okay, the correct answer is false low results due to antibody excesses. Let me explain it in a simple way. Actually, in proton effect, having too many antibodies prevent the antigen from cross-linking, which result in reduced signal and lower measure concentration. All right, I hope it's clear. Let's move on. Okay, the next question here. If enzymatic assay incubation temperature is increased beyond the optimal range, the most likely outcome is what will happen? The correct answer here is decreased absorbance due to enzyme denaturations. In simple terms, enzymes work best at the certain temperatures. If the temperatures gets too high, so the enzyme lose their shape and stop working properly, leading to decreased signal in experiments. All right? All right, that's all for today. Please don't forget to like, comment if you have any questions, share, and subscribe to our channel to receive all the latest lecture. I also encourage everyone taking the test to share your question with us so that everyone can benefit. Okay, that's all. Salam.